Good afternoon, everyone. Thank you for joining us. My name is Claire. I'm a contract librarian on assignment here with NOAA Central Library, and I'll be your host this afternoon. This seminar series is offered through the NOAA Central Library Seminar Program, which provides an educational forum for the presentation of ideas, research updates, and news in support of NOAA's mission. This is the third in a 5-part series, and the next in this series will be on Wednesday, September 29th at noon. Before we start, I have a few technical tips for attendees. Just so you are aware, you are muted and you are not able to unmute. However, if you have a question, you can type it into the question panel, and we'll get to those during the Q&A at the end of the presentation. If you're having any audio or visual issues, if you can't hear the speaker or see the slides, please try logging off and logging back on. That tends to solve most issues in GoToWebinar. And as a reminder, this is being recorded. If you'd like to revisit this seminar or view previous seminars, you can find them on the library's YouTube channel. Now I'm going to turn it over to Lisa, who's going to introduce our speakers today.
Hi, everybody, and thank you for joining. As you mentioned, the third in our 5-part series that is discussing and highlighting some of the significant findings of researchers during and since the 2010 Deepwater Horizon oil spill in the Gulf of Mexico. We wanted to put together this series of seminars that are highlighting our findings, showing the effects of oil on cetaceans that have been exposed to oil during and since the Deepwater Horizon natural incident occurred, and as part of and following on the natural resource damage assessment. There's a comprehensive research initiatives beyond the damage assessment, including the GOMRI research initiatives. And we just wanted to bring together a series of experts to talk about their findings and to help advance our ability to plan for, respond to, and assess for the impacts from marine oil spills. So with that intro to the series, and with an understanding that we've got 2 more coming up in the series, I wanted to introduce today's talk, which is entitled The Population Consequences of the Deepwater Horizon Oil Spill for Pelagic Whale and Dolphin Species. And we've got 2 excellent speakers. It's going to be kicked off with Len Thomas with the University of St. Andrews, and Tiago Marques with the University of, both with the University of St. Andrews. And I hope I didn't butcher your name too badly, Tiago. But you guys are really going to enjoy this talk, and I'm going to turn it over to our experts and stop rambling and let you get to the meat of the presentation. Thanks, everyone.
Thank you very much, Lisa. So I'll go first, and then Tiago is going to take over about halfway through. So let's get started then. Okay, so I'm presenting this talk on behalf of a very large number of people as part of the CARMA project, the Consortium for Advanced Research on Marine Mammal Health Assessment, which was funded by GOMRI. So you can see some of the people involved there on the slide. And in particular, this part of the talk comes from one component of the big project, which is the integrative modeling component, which comprises Tiago and me, but also these other people that are listed on the right here: Cormac Booth, Lance Garrison, Ryan Takashita, Patty Rosell, Keith Mullen, and Laurie Schwacke. And you can see us in our usual mode there on the right, which is sitting on a Zoom call, scratching our heads, talking about our population modeling. Okay, back in time, the Deepwater Horizon oil spill occurred on the 20th of April. As we know, killing 11 people, injuring 17 people, and starting the largest oil spill there in American history. The oil spill resulted in the death of many cetaceans, some of which washed up on shore and triggered an unusual mortality event. Many more strandings were observed than usual, which occurred over the next 4 years. And concern about the environment and the consequences triggered a natural resource damage assessment headed by the federal government, which took place during the 5 years following the spill. On the top here, I'm showing some field studies. There were studies of the stranded animals. There were hands-on health assessments of the bay, sound, and estuary dolphins that it was possible to get to, mostly in Barataria Bay, which is just on the west of where the Mississippi comes out into the Gulf of Mexico. And there were also photo ID studies and biopsy studies. The photo ID studies are focused on getting population estimates and estimates of population of survival rates over time. And these studies fed through into the stuff on the bottom here, which was a series of population assessments which formed the damage assessment. There was one for the Barataria Bay dolphins being the best studied Bay, Sound and Estuary bottlenose dolphin population. Also, there were studies for the other Bay, Sound and Estuary and the coastal bottlenose dolphins. And then there was a study on the pelagic stocks. There was a bunch of questions left unanswered at the end of that work, principally about the long-term effects. We had to make some guesses about what was going to happen in the future. And we put in a proposal that was accepted to GOMRI to form an ongoing study, which took place over the subsequent 5 or 6 years up until now, which involved more field studies. So that's more health assessments of dolphins to find out what was happening to their health in the longer term, follow-up photo ID to look at survival, and also an expert elicitation process where we put together in a very formal way experts to come up with distributions on parameters, population parameters that are not known sort of formally. We couldn't get formal data analysis to get distributions on them. There's a structured process for doing that, and we took advantage of that in this second part, in this CARMA study. And that resulted in a series of updated population assessments. So one on bottlenose dolphins in Barataria Bay. Again, the other dolphin, bottlenose dolphin, Bay, Sound and Estuary and coastal populations, and the pelagic populations. And if you were lucky enough a month ago, no, sorry, that would be 2 months ago, to be in the same place that you are now, watching the screen, you will have seen a NOAA library talk headed by Laurie Schwacke, who is head of the CARMA consortium, with some co-presenters, including me, on the Barataria Bay bottlenose dolphin update. And if you weren't lucky enough to be at that, then it's on YouTube, so please avail yourself of it. And we're going to today talk about the pelagic stocks, so that's where we fit in. We're going to present our findings on the updated damage assessment effectively for the pelagic stocks. There are 22 recognized stocks in the US EEZ, US Gulf of Mexico, and I'm listing them here in maybe a slightly funny order. So at the top here, I've grouped together 6 pelagic dolphin stocks. These two are on the shelf, so the shallower water. So pelagic means sort of surface feeding. I've grouped them here by functional group. This is surface feeding animals. These two are mostly on the shelf. And then these other 4 species forage in deeper water, but mostly restrict their foraging towards the surface areas. Mesopelagic divers, the next lot, of which there are 7 stocks. Mesopelagic means that they forage in the mid-water, at mid-depths. And then there are 2 stocks here that are deep divers. They forage at very deep beyond where the light reaches in the bathypelagic zone, sperm whale. And just to note here, I've grouped together 3 beaked whale species into one analysis. We didn't have enough data to do them separately. I've also grouped together pygmy and dwarf sperm whale into just a Kogia group. So all in all, we've done this damage assessment for 14 different taxonomic units, most of which are single stocks, but some of which are grouped. There are 4 more stocks here that I've listed at the bottom that we didn't analyze. There's just too few data for them, or not enough of them, that we felt we could do anything reliable. And for those of you who are into British whale, or now called the Rice's whale in the Gulf of Mexico, I'm sorry, but we didn't do that one. So if that's your bag, you might want to stop listening now to avoid disappointment. Okay, so these are the stocks that we looked at. The first question, I guess, that's relevant is, for each stock, how many were exposed to the oil? This is a satellite photo taken on the 24th of May, showing the surface oil. And what we relied on was a paper by McDonald et al. 2015, I'm listing the paper here, that covered the period 24th of April to 3rd of August 2010, that did a detailed analysis of oil coverage in 2 sort of categories. One was a very thin sheen of about a micrometer or so, and the other was a thicker layer of about roughly 70 micrometers. And what we wanted to do was we wanted to basically define a zone where the cumulative oiling was as much as or more than Barataria Bay, because we're going to do our population assessments building a lot on what we found for Barataria Bay. So we want to define areas where the oil was as bad as Barataria Bay or worse. So we cut the Gulf up into 5 by 5 kilometer cells, and we looked for areas where, according to this paper, the cumulative oiling was greater than 38.5 meters cubed. And this is what we saw. So, whoops, sorry, I've just pressed the wrong button there. Hang on just 1 second. Oopsie-daisy. There we go. Just let me whiz back a bit. Sorry about that. Something popped up unexpectedly. Okay. So this is what we saw. So the hatched area here is the oil coverage above that threshold. I'm also showing here, just for your information, the shelf boundary, which is this slot here, and the oceanic boundary. So you put those two together, that's the area that we're talking about, that we're concerned with cetaceans in. And then this hashed area is the oiling footprint. Okay, the next question is how many cetaceans were in that oil footprint and what proportion of the population does that represent? And to look at that, we levered heavily on this paper, which is written by Jason Roberts and colleagues at Duke University, where they took an enormous amount of line transect survey data, both from aerial surveys and shipboard surveys, and they analyzed it and fitted spatial density surface models to those by taxon, where they looked at an enormous number of covariates and predicted based on those covariates what the density was as a function of covariates. They had two types of models, and we use what they call their climatological model. So this is using long-term average covariates such as eddy strength, chlorophyll, things like that. And they produce those covariates over 8-day periods covering 46 8-day periods covering an entire year. So we used their predictions for each of those 8-day periods, and then we took an average to get the average density surface over the year. There's uncertainty on these models, of course. They're models, they have variance associated with them. And so to represent that, we sampled from the model 1,000 times, and we made 1,000 predictions of how many, what the population size was, and also what proportion of them were in the oil footprint. So here's an example for sperm whale. So this is the model here. Density is a function. They did model selection, and they found in this case that 3 variables affected density. The S there means a smooth, so density is a smooth function of the log of depth, the log of the maximum eddy kinetic energy, and the chlorophyll A concentration. I'm just showing the relationships here along the bottom. And this picture here is of the average density surface. And I've superimposed the oil footprint on top. So you can see that sperm whales are concentrated in this area here, right in the middle of the oil footprint, and also this area outside. And so if you sample from that 1,000 times, you get something like I'm showing on the right here, where the y-axis here is the proportion exposed, the x-axis is the population size, and there's 1 dot for each of those 1,000 samples. And so sort of looking along the x-axis direction, this is the distribution of population size. So the average is 2,561, and the confidence interval on that, you can just look at the lowest 2.5 and the upper 97.5 gives you 2,117 to 3,094. And then looking along the y-axis, this is the proportion exposed, 0.19, so 19% on average, somewhere between 17 and 22. Okay, so that's sperm whales. When you do that for all species, all of the 14, stocks that we looked at, you get something like this. So there's sperm whales, so the proportion exposed around 19%, the population size, what was that, 2,000 or so. And rather than go through 14 of these all the way through this talk, I'm going to pick out 2 more just for the purposes of illustration. So I'll pick out this one up here, that's the stock with the highest proportion exposed, which is spinner dolphin. And I'll also pick out this one here, which is a really high population size. So that's a bit of a contrast, a median proportion exposed, and it's the shelf stock of the bottlenose dolphins, as opposed to the oceanic stock. So let's just have a quick look at those. So spinner dolphin looks like this. There's actually a fair number, although it doesn't show very well in this picture, in this area around here, which is what gives them the largest proportion exposed of their population. There are really none of them around here. So it looks like about 38% of the population are estimated to be exposed between 22 and 56. and the population size of 16,000 with quite a wide confidence interval on that. And then the other one I said I'd show you was shelf population of bottlenose dolphins, which is here. And they were the ones that have one of the largest population size. So about 65,000, of which we're estimating about 20% were exposed. Okay. So how do we quantify injury overall? What do we do? So the first thing we did was we need a sort of baseline scenario. So we come up with a population dynamics model under baseline conditions. And what we did for the pelagic stocks was we assumed the population is at carrying capacity. So whatever that initial population size is, that's just what would have carried through if there hadn't been the Deepwater Horizon oil spill. Of course, this is an approximation to reality. We then took our population dynamics model and we ran it again, assuming some degree of injury, so an injured population, and I'll talk about how we did that in a second, but that creates an alternative scenario that is the closest approximation we have to what we believe happened. And then we summarized the difference between these two using 3 metrics. So the first one is the number of years until the population gets back to 95% of what it would have been if there hadn't been the oil spill, and we're calling that years to recovery. The second is the maximum proportional decrease. So this is the biggest difference, proportional difference between the baseline population size and the injured population size. And the third, this is my personal favorite, is lost cetacean years. So if you add up the difference between these two trajectories for each year, then that's the number of cetacean years that aren't there that would have been there under baseline conditions. So it's the sum of the baseline population size minus the injured population size. Please feel free to ask afterwards why that one's my favorite. Okay, the baseline population model. In a way, we don't really need this, right? Because we're just projecting forward based on the population that was there before, but we do need a population model that we can then perturb under the injured scenario. So we constructed a population model, and it's based on what we did for Barataria Bay dolphins with some tweaks. The Barataria Bay dolphin model is a deterministic model. That means that there's no randomness in it. For a certain set of starting values, you always get the same result. And I should have said on this that we repeat this process many, many times with different parameter values in them sampled from our uncertainty on the input. So uncertainty on population size, but also uncertainty on all those population parameters. And the population parameters here are, we're assuming that there are 60 ages for dolphins at least. This is females here, this is males. And then these arrows show that, for example, age 0 females go to age 1 females, and this is female age 0 survival rate. That's the S there. Female age 1 survival rate and so on. Once they get to a certain point, then there's an arrow that goes back here, and this is producing offspring. So this is the fecundity rate after they've reached sexual maturity. They produce offspring, half of which are male, half of which are female. The population model that we have is a density-dependent model that assumes a carrying capacity. So what that means is that when the population is really small, it grows, but as it gets bigger, the rate of growth decreases until it reaches some stationary value, which is the carrying capacity. So that's the value that the population would be if you didn't perturb it at all. And the slowing down is caused by the birth rate going down, the fecundity. So this fecundity here is a function of how many animals are in the population. The more animals in the population, the lower the fecundity, and that causes the population to level off. Okay, that's dolphins. All the other populations have a rather similar sort of reproductive biology and life strategy. So all the cetaceans that we're looking at have relatively long lifespans, relatively low reproductive rates. In population ecology, people used to refer to this as K selection, as opposed to, for example, locusts, which have very short lifespans but very high reproductive rates. So they're certainly similar, but they're also, we know there are differences between the populations in their lifespans, reproductive rates, and so on. So we need to tweak this model to produce something that would be suitable for each of these other species, in the absence of lots of information about all these demographic parameters. And we did that based on scaling by the duration of gestation. Gestation is the time between becoming pregnant and giving birth. There's a number of other options we could have done. We could have scaled by maximum lifespan, for example, but those other options that we considered, the parameters, for example, maximum lifespan, are not known at all well for these animals. whereas gestation duration is known fairly well for all of these stocks. We did a literature review and we came up with these values here. So this is gestation duration on the y-axis and species on the x-axis. So the spinner dolphin has the shortest gestation of all of the stocks that we considered. The sperm whale has the longest, and this is the bottlenose dolphin, which was the subject of our original Barataria Bay model, somewhere in the middle. So we used this gestation duration to scale the population parameters. And I'll give you one example, which is survival. So how did we scale survival? So this is the survival model that we used originally in the Barataria Bay population. So, and I apologize for those of you who don't like formulae, please bear with me just for a minute. Those of you who do will no doubt enjoy this. So this is the hazard of an animal dying at a particular age. So think of it as your risk of mortality, your risk of dying. And it's a function of these things here. And there are 5 parameters here, beta 1, 2, 3, 4, and 5. And they were derived from an analysis of strandings data on bottlenose dolphins, not in unusual mortality conditions, but just under normal conditions. And so these parameters, 2 of them, this beta 2 and this beta 5, are multiplied by A, which is age. So mortality at age A is a function of this lot, which is a function of age. So there's a minus sign here. So this term is going to go down as you get older. So this reflects childhood mortality getting less of a problem as you get older. This one goes up as you get older. So this reflects the fact that your chance of mortality goes up as you get older, particularly for much older ages. And there's a C here. That C is how we took account of gestation duration. That C is the ratio of gestation duration for dolphins divided by gestation duration for whatever the target stock is that we're interested in. So the effect of that is shown here in a survival curve. So on the y-axis is probability of survival. This is for females and the bottom one's for males. And this middle green dashed line here is showing how survival goes up and then down as you get older if you're a female dolphin. And you can see that for the spinner dolphin, that is pulled to the left. So your survival, you're not going to live as long effectively. Whereas for sperm whales, your survival rate stays higher for longer. And so sperm whales are going to live for longer. The sperm whale population is going to take longer to age. And so effectively, this ratio is just pushing and pulling that hazard of mortality, which changes the survival curve. We did something very similar for fecundity that I won't have time to go into in detail. Okay, that's baseline. What about the injured population? So for the injured population, we grouped them into 3 different groups. So there's these guys here who are exposed animals who recover at some point in their lifetime. These are exposed animals that never recover, and they eventually produce this lot here, which are animals that were born after the oil spill. And there's a parameter in the model for the proportion of animals that recover that we derive for bottlenose dolphins. There's this thing here, which is the reduction in survival, SF, reduction in survival that's caused by the oil spill. And that's a parameter in the model. And then there's this one, which is the reduction in, this RF thing here, reduction in fecundity that's caused by the oil spill. How did we get those? There isn't formal information to go on. We had some information about reduction in survival for Barataria Bay bottlenose dolphins, but we want to say, what's that going to be for these offshore species? So we convened an expert elicitation of these people who are listed here, some of whom may be listening right now. So thank you very much indeed for your time and your efforts if you were. And we asked them, given what we know about Barataria Bay dolphins, what would you say should happen for 3 different groups, for these pelagic dolphins, for the mesopelagic, and for the bathypelagic? And these are the resulting distributions. Effectively, what they said was that for the pelagic dolphins, things should be slightly worse than for the Barataria Bay dolphins, with quite some uncertainty on that. And then for mesopelagic divers, it'll be rather worse because they dive deeper, they have breath hold that they need to do, and so their lungs are likely to be more sensitive to oil, and then even more so for the bathypelagic divers. And you can see that the uncertainty is going up as well. And then, so that's for survival. And we did something very similar for the reproductive factor as well. We basically looked at the ratio of the survival factor for these species versus dolphins and scale things appropriately for the reproductive factor. There's the formula there. I won't dwell on it. Okay, so to summarize then, these are the model parameters overall. So in the baseline population, there's, I've colored them. So a green one is it's species specific. An orange one is that it's one of these ones that we get from gestation duration from the bottlenose dolphin model, and then scaling by gestation duration by species. And then the blue ones are these functional group specific, like for example, the survival reduction I just talked about. Okay, so the baseline population is initial population size. And then for survival, there are these age and sex specific survivals, which come from the Barataria Bay model scaled by gestation duration. For reproduction, there's an age at sexual maturity, which comes from the Barataria Bay model scaled by gestation duration. There's maximum fecundity, so how much is fecundity going to be, what's it going to be at very small population sizes. Something to do with density dependence, which we assumed is the same for all species. And then something that gives you the carrying capacity that we're at right now, which was estimated given all of these other parameters and the population size that we assumed. And then for the injured population, you have exactly the same parameters plus some injury parameters. So what proportion of the animals were exposed, I talked about before. What's the survival reduction factor? That comes by functional group from the expert elicitation. What's the reproduction reduction factor? So that comes from the expert elicitation, and it's also scaled by gestation duration. And lastly, what's the proportion of the exposed population that recovers, which comes from that dolphin study? Okay, I'll stop there and hand over to Tiago.
Very well. So I guess this is where I take over. I'm now being given permission to share my screen. So hopefully you are now seeing a slide that says results.
Yep.
Very well. Okay. So I take it from Len here, and we focus on the results. In fact, the heartbeat is actually explaining what we did and how we got there, and Len's done that very well already. So the results are really just a bunch of numbers that we try to make sense of next. We will sort of use the spinner dolphin, the sperm whale, and the shelf stock of bottlenose dolphins to guide us through the results, kind of as Len did in one of his slides. So, you know, after getting the computer to run for a while and simulating these thousands of iterations for each one of the taxonomic units considered, this is a summary of the injury metrics. Come on, I do not want to leave the webinar. This is a summary of the injury metrics we got per taxonomic unit that we considered. So with respect to maximum proportional decrease and a closely related measure of years to recovery, which, as you recall, we defined as recovering to at 95% of the initial population size, we see that we had 6 taxonomic units with more than a 5% decrease. I mean, naturally, these are the ones for which the median time to recover is actually larger than 0. The worst impacts were in this case for the spinner dolphin, Sternella longirostris, and the sperm whale, Visitor macrocephalus, with a predicted time of recovery larger than a decade. The shelf bottlenose dolphins, Tursiops truncatus, shows here midway through the table, while Clymene dolphin and the Atlantic spotted dolphins appear to be the least affected stocks for these 2 injury metrics. Why is the slide not moving? There we go. So while the numbers on the previous slide might seem to represent relatively small impacts, impact can be measured in many ways. And if one focuses on lost cetacean years, over 100,000 cetacean years were lost across the 15 taxonomic units, with the main contributors being the shelf stock of bottlenose dolphins, as well as Sternella attenuata, the pantropical spotted dolphin, with over 30,000 lost cetacean years each. And the spinner dolphin comes third with over 15,000 lost cetacean years, while the sperm whale comes halfway through the table with about 2,300 lost cetacean years. So if one switches gear to focus on each one of the species, one can get a bit more appreciation about the variability in the results. So do not be fooled by the fact that all the lines seem to vastly overlap each other, while the initial population size is, reflecting the uncertainty around the initial population size. They vary a lot, but each one of those lines in the baseline scenario is paired with a line in the impact scenario, and hence the effect might be small, but it's present on each one of those lines, and therefore it sort of transpires at the end. So note that the variability in the median years to recovery, allowing us here to understand better why before the median for the bottlenose stock was 0. While more than half of the populations do not show a maximum proportional decrease above 5%, many do, and therefore for those there's a distribution for years to recovery here, for which the upper 95% confidence interval is actually larger than a decade. The same pattern can be seen to a lesser extent for spinner dolphins. We still see that there's a fair number of realizations where the maximum proportion decrease was lower than 5%, leading to 0 years of recovery. But now for most of the realizations, we are above that value. And so the upper confidence limit in years to recovery is actually over 2 decades. So regarding the sperm whale, we see that the pattern is similar, and the median years to recovery are close to a decade, considering the upper limit of the confidence intervals to represent values one might not want to dismiss. And it's actually not that unlikely that the maximum proportional decrease could reach up to 10% of the population, and that up to 2 decades might be needed to recover to within 95% of the baseline population. Now, there are a lot of species, lots of parameters, 3 injury metrics, so it's a bit hard to convey all the results in an integrated way. So we chose to present them here by looking into what are the main drivers of the injury metrics using the species as if they were replicates. So considering a very simple regression analysis, we modeled the value of the injury per species as a function of the input parameters. And we use the mean of the injury metrics as a response variable and the mean value for the input parameter, input parameters, so initial population size, proportion exposed, gestation duration, which as Len explained was used to scale survival and fecundity, and survival reduction as explanatory variables. With a simple model selection procedure, removing non-significant terms from the model, and the output of these models allows us to describe what drives the key components of variability in the injury metrics. So when we consider the maximum proportional decrease, we can see that the proportion exposed actually explains 91% of the variability. And then survival reduction explains some further 6.1%, and gestation duration still helps to explain 2.1%. Once we do that, we see that, the model that includes these 3 quantities very well explains all the variability in maximum proportional decrease, and only a residual amount remains to be explained. On the other hand, when you look at lost cetacean years, it's only when you look at years to recovery, it's only the proportion exposed and the gestation duration that seem to affect the outcome, and about 74% of the variability is induced by proportion exposed and about 11% by gestation duration. And so there's about 15% of the variability that's not explained by any of these 2 measures. Finally, when you look at lost cetacean years, we see that there's 2 drivers of the variability. Most of it comes from the initial population size, as would be expected given the nature of this metric, with about 88% of the variability explained, and then the proportion exposed of the population explains some further about 5%, and therefore about 7.5% of the variability remain to be explained by the model. So overall, these results were kind of consistent across species, but we also wanted to evaluate the sensitivity of the model to the different parameters involved in the analysis. And so, oops, sorry, what's going on there? So we wanted to do this sensitivity analysis, and we considered both an uncertainty analysis and an elasticity analysis. Uncertainty being, how much does the variability in the input parameter induces variability in the outcome, and the elasticity tells you a little bit about how changing each one of the input parameters by a small amount might induce changes in the results. So the approach was, for each species, for each input parameter, fix all the other input parameters at their mean, repeat this many times, and then just look at the resulting injuries to evaluate these different uncertainty and elasticity components of sensitivity. So an example, so here on this slide, we kind of illustrate further the procedure for the sensitivity analysis using the proportion exposed as an example parameter and the sperm whales as an example taxonomic unit. Remember, we had the distribution of the animals in space and the oil footprint on the left top corner, and then we actually resampled parametrically from the spatial model to have 1,000 realizations of the initial population size and the corresponding proportion exposed. Now, given that, and fixing all the other parameters at their mean, while kind of using many resamples, in this case 10,000 of the proportion exposed distribution, we run the model 10,000 times and evaluated the 3 injury metrics in each one of these times. And that's what's represented in the plots to the right. So actually the variability on the y-axis represents how much each of the injury metrics has changed as a function of the current input parameter variability. Here the proportion exposed, and that is what we refer to as uncertainty associated with the proportion exposed. On the other hand, the slope of the relationship between the parameter and the injury represents how much changing a small amount of the initial parameter induces a change in the corresponding injury metric. And that's what we call the parameter elasticity. So this is an example result for the sperm whales, because as you might imagine by now, there's 15 taxonomic units and tens of parameters, so that's hundreds of comparisons, but and values. But overall, we concluded that the main drivers of the variability, so uncertainty in the initial parameters, was being introduced through the baseline survival, the density dependence component, and the survival reduction. And these were responsible for most of the uncertainty. In terms of elasticity, survival reduction, density dependence, the proportion exposed, and the maximum fecundity were those parameters that by changing small amounts would lead to larger changes in the injury metrics. And so that gives us a notion of which ones are important and which ones might lead to changes in results if they change a bit. So those were briefly the results we got. Small discussion to end. So while the ecosystem impacts have been severe and widespread, their quantification is actually necessarily hard to do. And so here we focused on pelagic species for which said quantification is possibly even harder, with most species lacking the relevant information required to do so. We used expert, we kind of tried to include all the best available information in our models, and we filled the remaining data gaps we replaced them with some assumptions. Expert elicitation allowed us to include information that was not directly available as traditional data, which was very helpful. And as noted, we have scaled, the strong information available about survival for bottlenose dolphins to all these other taxonomic units based on gestation duration. And in particular, we did not account for variability in gestation duration. We considered a number of alternatives, as Len said, but gestation duration was considered the best since there was relevant information available for all taxonomic units, and it was believed to not vary much geographically, which might not be the case for other alternatives, which would make the use of information collected elsewhere much harder. And all these choices naturally have implications on the results that could be discussed at length. In this model, density dependence is key for recovery, because it's that dependence on density that affects the fecundity that allows us that the population can recover. And so without this component, injury would be permanent. So there would be no way to recover from it. And so that's a fundamental aspect of the model that one needs to consider when, putting these results into perspective. Finally, it's also important to say that we are quantifying impact on populations via these direct pathways, but overall impacts could actually be much more convoluted and hard to evaluate and assess, because they could include other indirect pathways. Some prey for one of these species could be specially affected, and that could have its knock-on effects on the population itself. And so overall effects might be considerably larger than what one is quantifying here. And also, it's important to note that we've used different metrics to quantify injury, but these different metrics actually quantify slightly different aspects of injury. And so there are really no perfect population level injury metrics, and no single metric can capture the full injury. So we've used these, but others might be considered, and each one of them tells us a little bit something about the process. And just as an example, so you can see how results could be sensitive to some of these. If we change the years to recovery to be the number of years required to achieve only a 2% decrease with respect to the threshold instead of a 5% decrease, then this could lead to considerably larger years to recovery, which could be seen as if the recovery would take considerably longer to occur. Also important to note, I guess, is that we have established that the proportion exposed is the most important injury driver and seems to be relevant for all the injury metrics that we've used. But we should recall that we actually assumed a static model for exposure, in the sense that animal movement was ignored. We had the oil footprint, we had the distribution of the animals, and the overlap gives us the proportion exposed. And if animals move a lot and they have different reactions to oil, then we could actually see the proportion exposed go up or down, which would have knock-on effects on the injury metrics that we've computed. Finally, I would like to just add a note to say that a research paper is being submitted soon on these results, and all the results as well as code that you would need to reproduce yourself this analysis will be publicly available on some repository. And this actually means the analysis is transparent, you can easily reproduce, and it's updatable in face of new information. So a few take-home messages just to conclude. So the Deepwater Horizon, as we all know, has had large and potentially lasting impacts on many components of the Gulf of Mexico ecosystem. And despite these impacts being large and widespread, quantifying the impacts is extremely difficult, especially when facing the absence of baseline data. We have here quantified many thousands of lost cetacean years across the multiple taxonomic units considered. We note that the model considered focus almost exclusively on direct health impacts induced by oil-associated chemicals, and therefore this could correspond to an underestimation of injury if other indirect pathways lead to accumulated negative effects on the species considered. And as an example, which I've noted, if the preferred prey of a particular stock was severely impacted, then knock-on effects would follow. We saw that all the key model parameters considered had an influence on the injury metrics, and that the proportion of the population exposed was, not surprisingly, responsible for explaining most of the injury, having an impact on all injury metrics considered. So while it is possibly that the recovery, here quantified as back to 95% of the baseline population, might have happened already for some species, it is also not unlikely that for some of the species, that recovery will, hopefully, have to continue for a while. So the model presented provided a way to evaluate injury in this specific setting where baseline data was not available for many species, and we believe we produce results which are reasonable and could be used to support restoration planning. And finally, we note that none of these results need to be cast in stone. The models have been developed and implemented within a reproducible research paradigm, and hence if new information about any of the relevant components in the model becomes available in the future, it will be straightforward to adapt the existing framework and recalculate the estimated injury. And I guess that takes me to the last slide, where I just really wanted to acknowledge again that this is a really, really large team effort. So there's many CARMA partners that we would like to thank, and too many collaborators to name anyone in particular, but they are all very appreciated for the help that they provided, and acknowledge the funding that we received from the Gulf of Mexico, GOMRI. So that's it, folks. If you have any questions, I think this will be the time to have them.
Yep, thanks, Len and Tiago. If you have any questions, please enter them into the question panel on GoToWebinar. It looks like we do have one here. Can someone explain what lost cetacean years are considered to be?
I guess I'll have a shot at that one since I tried once already. I'll try again harder. So it's a good question. It's a slightly difficult concept to get your head around, so let's try. So if we think about how many animals there would have been each year, and then we think about how many animals there were under the oil spill, we think that there are less animals than there would have been. So let's say in year one, there would have been 100 animals, but we think that there were 80. So that's 20 animal years that aren't there because of the oil spill. And then in the next year, there would have been 100, but let's say now there are 70. So that's another 30 animal years that don't exist that would have done. So add those two together, that's 50. And keep doing that all the way through those trajectories. Add together the number that are there, the number that would have been there minus the number that are there. Add that lot up. That's the number of lost cetacean years. It's the number of years of life of marine mammals that are not there, that would have been there if it hadn't been for the oil spill. So you can think about it very loosely as that shaded area. It's the difference between the two curves.
Thanks. So I'm gonna wait just a moment here, see if there are any more questions.
In case not, I'm going to take the opportunity to explain why I like that metric, because there are Tiago said there's lots of different ways to calculate damage. And also, as he said, the problem with the one that a lot of people like, which is how many years is it going to take until they recover, is that it's super sensitive to what you mean by recovery, because these two curves get closer and closer and closer together over time, but it takes a long time before they actually touch. So it could be 100 years or 200 years in this model before they're actually exactly the same. And so we end up saying, okay, what about when they're 95% similar? So when there's been 95% recovery. But if you made that 95, 96 or 97 or 98, you get a really different answer. So I don't like that metric because it's super sensitive to what you choose to mean by recovery. And then the problem with the maximum proportional decrease is that that's only one tiny element of the damage. So, how much did you knock the population down to is just one thing, but really it could be many, many, many years before they're back to where they were before. And so I like this lost cetacean years metric because it's pretty robust. There's no arbitrary decision about what you mean by recovery. And it summarizes the damage, I think, in kind of one number.
Great. I actually did have a few people ask about that, so thank you. Our next question is, how would you integrate ecosystem indicator in the model?
Tiago, do you want to do that one or shall I?
How do you integrate ecosystem indicator in the model? I don't quite fully understand the question, to be honest. Actually, and I'll take the opportunity that I'm mumbling to say, I just realized I was not transmitting video for the entire duration of my talk, apparently. So
it's a
sorry
shame to have missed out on your face for that time.
I'm sure that's not it, but anyway, yeah, I was waving a lot, but anyway.
I'm guessing, so one thing that we've assumed in this model is this idea of carrying capacity. So the sort of number of animals that would have been there without injury and the number of animals that this returns to given the injury, that the trajectory returns to. And you can think of an ecosystem, one use of an ecosystem indicator would be to say, how healthy is the ecosystem? In other words, how much food is there for these whales? And so you could, if you had some indication of something to do with the ecosystem, like some indicator of the fish that they feed on or at these different water depth layers, then you could incorporate that by changing the carrying capacity. And that would let them to recover to a different number. So that's one way I could think about incorporating an ecosystem indicator.
Thank you. I'm going to wait just a minute here to see if we have any other questions. All right, oh, here we go. I do have a question for Tiago. In the modeling of the drivers of max pop decline, do the drivers vary by species?
Yes, they do vary by species. So what you use is the value, in essence, the mean value for each one of the drivers that got into that species, and then you predict the injury of that species, of each one of the species, as a function of those drivers. So yes, they varied by species.
Just to follow up a little bit on that as well, there's one thing that people may be thinking, which is that if you can predict the damage by simply knowing, so for example, we said for years to recovery that, no, I won't say that one, lost cetacean years, my favorite. We can predict it, around 90% of it by initial population size and proportion exposed. So you may say, okay, well, those two you had at the beginning of the talk, why did you bother with the population model? And the answer is we can predict the relationship between lost cetacean years across those species, so the slope of the line effectively from those two things, but we don't know what the intercept is without the population model. So given the population model, then all we need to know is those two things to have a good idea about what the injury is going to be, but you still need the population model in the first place.
All right, the next question here is the assumption that POPs were at carrying capacity valid. They could be kept below K by disease or other factors.
Yeah, we debated that one a lot. Shall I talk about that, Tiago?
Sure. I mean, we needed to make some assumptions to have a way of starting the model. And, you know, if you want to evaluate injury, you have to make up a reality that we never observed, right? Because that would be what would have happened in the absence of oil, and we just don't have it. So the way we did it was by assuming that the population was effectively at carrying capacity. But I guess for each one of the different species, you could discuss whether that's valid or not. And we did. And in particular, I remember that Len did a fairly intricate analysis about whether sperm whales would be, for example, at carrying capacity. And we concluded that it seemed like a fair assumption, but, you know, it depends a lot on the biology of each one of the species. I don't know,
I would
Len,
say,
if you want to comment.
yeah, I would say for the, I mean, if they're kept at below what we might call below carrying capacity by chronic disease in the population, then that just means the carrying capacity is different, right? If it's chronic and ongoing, that just means that there are fewer animals in the long term, and so the carrying capacity is lower. So I would argue they're still at carrying capacity. It's just lower than what you think it is. So animals being below carrying capacity might be because there's a sort of something unusual has happened at that time, or that there's an increasing threat from, for example, anthropogenic change. And the one, as Tiago said, that we thought there was, I mean, there's really no evidence when you look at the, for example, the NOAA population trajectories. There's no evidence of population change in, as far as I remember, any of the populations, although the uncertainty on those NOAA estimates is very wide. The one, though, that you might think from first principles is changing is sperm whales, because they were historically hunted in the Gulf of Mexico almost to extinction. And so they've been recovering since then. And so the analysis Tiago is talking about is I looked at the whaling records and did an analysis of what if there had been carrying capacity whales, they'd been depleted by whaling, would they have had time to recover to carrying capacity by now, by the time of the Deepwater Horizon oil spill? And I think the answer is probably yes. And I'd like to write that up at some point as a paper, just to include it in the sort of body of work around this. But so we don't really have any reason to think that the animals weren't at carrying capacity, although we do have to discuss what we mean by carrying capacity for sure.
Right. Do you have any ideas how to include animal movement in the models?
Short answer is no. So what you would expect animal movement to do is to reduce the injury to each animal, because they would move out of the area quicker, and so they would be in that, unlike the Barataria Bay dolphins that stayed there the whole time, they would be less exposed, but more of them would be exposed, because if they're moving randomly, then some of them will move into the area. So it's very hard to predict which way it would go. The only way you might think it would have less of an injury is if animals move out of the area because of the oil spill and don't come back and avoid the area. But even then, if the animals are moving out of the area, presumably they were there for a reason, which was that there was food there or something that they wanted, and so there's less available as they move out. And so there's no reason really to suppose the injury is any less. So I agree it's a very difficult topic, and I don't have an easy solution. Tiago?
No, you know, as you said, you can make it go one way or the other, depending what you assume for those movement patterns. And it's not just movement patterns, because even we don't know how most of these animals might move without the oil there. Then with the oil there, then who knows? And so it becomes even more hard to try to include that component. But I agree, it must be an important component, because, you know, from first principles, if I am able to avoid the oil, then I'm certainly going to have less of an impact than if I'm not and I just swim into it. And so I think movement is key there in some way.
All right, and I think this is going to be our last question. Do you have plans to use a similar approach to a different human impact?
That's a good question. So I really like this approach, and I think it has a lot of potential for other populations that have been impacted by human intervention. So it could be chemical oil spills, but it could be other things that are impacting populations. So this idea of assembling all of the information you have with a population model, unifying it together, and then sampling from the uncertainty on those things, gathering data. We can use the uncertainty analysis to target where the biggest uncertainties are affecting our model, and so try and reduce those uncertainties by gathering information. And we can use expert elicitation to formally put together diverse sources of information in a coherent and reproducible way. So I really like it, and I'm very open to other opportunities to use this kind of framework in other scenarios. We have one or two in mind, but I'm very happy to hear about any others that people think would be useful.
Yeah, and I'll just add a few words to Len's answer to say that one thing that I would like to do, time allowing, which there's never enough time to do things you would like to do, but anyway, would be to use this approach in a situation where we actually had real data for all the things that are involved, and then see how you could actually, do with an approach where you just start taking components of the data and pretending you don't have it to see if the approach still allows you to get close to the reality that you can actually have data and quantify directly. So I think that would be a nice way of further validating the approach, and that would be a cool study.
All right, that is all we have time for today. If we didn't get to your question, I will forward those to the speakers so that they can follow up with you. I just want to thank our speakers and thank you all for joining us. You will be able to find this presentation on YouTube later this afternoon. When the presentation ends, you will see a survey. If you could take the time to fill this out, we do use these surveys to improve marketing our library seminar service and to inform future seminar topics. Just a reminder, the next seminar in this series will be on Wednesday, September 29th at noon. Thank you again for joining us. Have a great rest of your day.
Thank you, guys.